Antibodies and vaccines, AQA Biology. Key terms. Antibodies are proteins with specific binding sites synthesized by B cells. Monoclonal antibodies are specific to one antigen. Active immunity is when the immune system makes its own antibodies after being stimulated by antigens that have entered the body. Natural immunity are infected under normal circumstances. Artificial immunity, vaccines and breast milk are examples of active immunity. Passive immunity, antibodies made by another organism. Vaccinations contain antigens that cause your body to produce memory cells against the particular pathogen causing diseases. Herd immunity, when a large proportion of the population has been vaccinated to make it hard for the spread of pathogens. Antibodies. This is the structure of an antibody. You have where the antigen would join, which is the binding site. So it would join here. Then you have the variable region, which is here. These will be different shapes depending on the antigen that is going to bind. Now you have the hinge region, which is this. You have the light chain and the heavy chain. Now the light chain and the heavy chain always are the same. It's the variable region that can be changed. Neutralization, which is when during an infection any effects are neutralized. Agglutination, fighting infection, which increases microbial elimination by phagocytosis, as large clumps of bacteria can be eliminated in just one pass. And opsonation, don't listen, you'd struggle with that word too, which fights infection by the pathogen being marked for ingestion and eliminated by a phagocyte. And these are the primary and secondary responses to antibody concentration. So primary would be you get the infection, there's a lag time, proteins are produced, antibody level rises, the infection is dealt with, and then the antibody level decreases. But a secondary response would be the antibodies are made again after the infection. It's a rapid response because of those memory cells that were present, a stronger response, and it's longer living, so it stays in the body longer after the secondary response. Which is why some people, for some diseases, when you go to get a vaccination, you have to get it twice because it makes the response rapid and more stronger if you were to get it. Vaccinations. There are two forms of immunity, active and passive. Active requires an exposure to an antigen. It takes a while for the protection to develop. Memory cells are produced and there's long-term protection. However, during a passive immunity, no exposure is required. The protection is immediate, but memory cells aren't produced and it's short-term. Now, what is the successful vaccination project? Well, it's economically available, which means just because you're less well off, you can still afford to buy it. There's few side effects. For instance, you wouldn't want in the case of thalidomide. I'm so sorry if I said that wrong. But that case whereby mothers were taking it as a morning sickness pill, and the baby, when they had their babies, they came up with deformities as a side effect of taking the drug. It's easy storage, and there's easy ways of taking vaccines, such as taking it through edible form or an injection and such. And it's accessible to everyone. Now, why would a vaccine not work? Well, sometimes there's failure to induce immunity in some individuals, and some individuals develop the disease immediately after the vaccination, so there's no time to build up a resistance against it. A mutation, different strands of the same pathogen, For instance, like which would be an example, the flu. Every year there's a different strand of the flu going around, which is the reason why you get elderly people that are more at risk to get a vaccination with what they suspect the cold flu will be this year. And then ethical reasons. In some religions, it's against their religion to take vaccines. Now, what are the forms of vaccinations? Well, you have live, which are, they're not dead, they're not harmful to your body, so your body can just fight against it. Dead, attenuated, which is weakened, toxoids, DNA, and edible. Ethical issues. Well, the use of animals is a common thing in which you probably hear about a lot. Side effects. Who should this be tested on? Because you do need a round of human volunteers in order to test the drug on. Health risks. If vaccines are so important, shouldn't they just be compulsory? And it's expensive. Now, I hope this was helpful to someone somewhere. And if you liked the video, then please like and subscribe. And the next video will be about HIV and monoclonal antibodies. Thank you for listening.